Hi, my name is Lauren, and I'm a third year undergraduate in the School of Natural Resources and Environment. These are the preliminary results for my undergraduate thesis. This project is a collaboration with the Department of Environmental and Global Health, the Department of Environmental Engineering Sciences, and the IFAS Fort Lauderdale Research and Education Center. Harmful algal blooms are caused by an overgrowth of algae in waterways. These are natural phenomena that occur worldwide in pretty much any type of waterway you can think of, but we're focused on a specific type called Carenia brevis that causes red tide events in the Gulf of Mexico. This is an example of what red tide can look like in the ocean. The health effects of red tide events extend to marine ecosystems and human health. For marine ecosystems, the die-off of fish and marine life are the main concern. This is caused by the algae delivering neurotoxins to the marine life, the decay of dead algae in the ecosystem, and the depletion of oxygen just because the algae is overgrown in the ocean. For human health, toxin containing seafood is an issue with red tide events, and respiratory irritation is also an issue, which is what this project is about. Respiratory irritation is caused by the aerosolization of algae in the air and can be exacerbated by wind direction and pre-existing respiratory conditions such as asthma. The mechanism by which HAB caused these adverse respiratory effects in humans is not yet known. Our hypothesis is that exosomes help to facilitate the immune response in lung epithelial cells. So when HAB are aerosolized, the elements of the algae make their way into our lungs when we breathe air. These lung epithelial cells that make up the inside of the surface of our lung react to the algae as a stress. And we think that exosomes are produced as a result of this stress and that they signal to activate alveolar macrophage immune cells, which eat the foreign objects, which are the algae elements, in the lungs when the lung epithelial cells respond to stress. So we are looking specifically at lipids that are contained in these exosomes to see how the lipids are related to the immune response. So here's our hypothesis written out. Again, we think that the elements from HAB under varied environmental conditions induce the secretion of lipid-containing exosomes from lung cells, causing changes in immune cell activation. To see how the abundance of lipids changes based on different algae treatments to lung epithelial cells, first we have to aerosolize the algae. To aerosolize the algae, we use a chamber on the top of black hole to expose algae to air. Then we take this aerosolized algae and expose lung epithelial cells to it. We then isolate the exosomes that are produced from lung epithelial cells in response to the algae, and then we use mass spectrometry to measure the relative abundance of lipids in those exosomes. So our preliminary data show that exosomes do in fact are produced as a result of the stress of algae in lung epithelial cells. Furthermore, we see that the lipid abundance in these exosomes is different based on the different treatments that we expose lung cells to. So the control is the background air of the aerosolizing chamber. The aged algae in green is algae that has been exposed to sunlight before aerosolization, and the fresh algae treatment in blue is algae that has not been subject to sunlight before aerosolization. So we can see here that these three lipid examples show different abundances in the exosomes produced by the lung epithelial cells based on the different treatments. There are differences between the control, the aged, and the fresh all across the board. There are differences between the control and the algae and the specific algae treatments. So our next steps are to go through thorough analysis of these lipidomics and to investigate functional impact of exosomes on immune response. Thank you.